All right, so thank you, Dr. McIntyre, for the introduction, and all of you to show up today. I have no disclosures. So what I'll be presenting later today at the in the session this afternoon is a concept of overcoming this signal, the "don't eat me" signal, of course, which is the wish of these lymphomas, and we will show data that this strategy actually overcomes the drug resistance that you see in these refractory lymphomas, and you get clinical remissions with quite minimal toxicity. So the way I think about the problem that we're trying to solve here in these lymphomas is drug resistance. So most of the time, of course, we're talking about chemotherapy resistance, but now we also think about it in the terms of these targeted agents and resistance to those. So with aggressive lymphomas, of course, the chemotherapy that they get can cure most of these patients. But when they do become resistant, when they relapse, the treatment options are quite limited. Now, in indolent lymphomas, the chemotherapy that we give doesn't cure the patient, and it is true that they have multiple treatment options, but it is also true that with subsequent therapies, the point of diminishing returns happens quickly, and the remission durations are much lower and shorter as you get to second and third line therapy. So any patient then that has lymphoma that's resistant to chemotherapy has a pretty poor prognosis, and the effective treatment options are quite limited. So all of these novel therapies, the entire goal is to both overcome the drug resistance, but also it has to have a safety profile, and that safety profile has to be such that it can be given to a large number of patients, at least in order to have the greatest impact. So the drug that we're talking about here, the novel agent, is something called HU5F9. So this is a humanized monoclonal antibody, and it targets something called CD47. So CD47 is the "don't eat me" signal. So this is, at least it's the pathway that inhibits the phagocytosis by the macrophages. So CD47 is on multiple cells, okay, throughout the body. But in cancer cells, it is coupled with an "eat me" signal. So there is an "eat me" signal, and in that way, that is the cell that would be eaten if you block this "don't eat me" signal, CD47. So rituximab is given with HU5F9. Now, this is important because rituximab, through its activity on the FC receptor, actually places an extrinsic "eat me" signal. So when you give these two things together, you're blocking the "don't eat me" signal, you're placing the "eat me" signal, and then the cell becomes susceptible to phagocytosis. It's already known before this presentation that this is effective. This has been published in the New England Journal of Medicine, so it is, at least the early phase 1 was published and shows that it's active and safe in patients with drug-resistant lymphomas. But what was not known before this presentation is how long that effect lasts. So this is a longer follow-up, and we actually will show you that the effects are durable and with continuous dosing. So the results of the study that will be presented, it's a large number of patients that have been presented already. It shows that remission can be induced. So by remission, I mean not only responses, but actually complete responses. So in patients that are, that have tumors that are refractory to chemotherapy, there's actually complete responses seen with these two drugs. And that can be seen both in large cell lymphoma, but also in indolent lymphoma. So both tumors are sensitive. The response rates are higher in indolent lymphoma, not too surprising, and the complete response rates are higher. And we'll show that. Also, with continuous dosing, we see that the side effects are they're quite. It's a tolerable drug, but it also is true that most of the side effects occur in the first month. So when you target this, the one cell of the body that is susceptible to targeting this is the aging red blood cells. So as expected, you will get anemia when you give the treatment. But we'll show that almost all that anemia occurs the first cycle, and then even though you keep giving the drug with continuous dosing, that hemoglobin rises, and the patients actually don't have anemia with continuous dosing. And we'll show you that the remissions are durable. So the early patients, the first 25 that were in the phase 1, have now gone on, and we have patients that are out past 2 years and still continue to get drug and have not lost their remission. And that's probably the most impactful finding of this presentation. So I think in conclusion, the way I put this together, this is a combination that has durable activity, and it also works in patients that already have this chemotherapy resistance, which of course is the goal to overcome. And the safety profile is remarkable. So these two monoclonal antibodies, the benefit here is then it can be used in almost all patients, so independent of age, and it's off the shelf. It doesn't need to be a procedure, this kind of thing, and that's where the most impact potentially could come. So these studies that we have to this point don't show us how it compares to other treatments. It's hard to compare phase 2 studies, but that is needed, at least in the patient populations that we see. And I think also, importantly, translationally, some of the correlatives that need to be done is we really want to have a better understanding of who benefits. So it doesn't benefit all patients. So one of the most important things is patient selection and trying to figure out who it is that benefits the most from this. But because the safety profile is so well, I don't want to say minimal, but it's certainly it's modest, then that means other agents can be added. And so that's an important thing for future research, is we can think about what agents should be added to this combination to sort of improve the outcomes. And with that, I'd like to answer any of your questions.
Thank you very much.
Hi, Neil Osterwell with Hematology News. So is this the same molecule as TTI 621?
It's not the same molecule. I'm not familiar with what. There are other agents that will be targeting CD47, both with antibodies or small molecule inhibitors. The CD47 actually will, the way it does its effect is it targets something on the macrophages known as SIRP alpha. There are agents that target SIRP alpha. So this pathway is being explored with a number of different agents. This is the first-in-class monoclonal antibody, so it's the furthest in development, this humanized antibody here.
I see, yeah, the one that I was asking about does target SIRP alpha.
Yeah, and so that's a similar concept, but it's targeting something actually on the macrophages themselves.
Okay, and do you think there would be a difference in performance? Or?
It's hard to know. I haven't seen all the data about these agents. I think it's fair to say that this particular pathway is exciting, and so a number of different agents are being developed, sort of similar to what you might think of with checkpoint inhibitors, PD-1 and PD-L1. You know, one's targeting the ligand, one's targeting the receptor. Those, of course, are inducing a T cell response, and here we're trying to induce a macrophage response. So but it's a similar concept in that there may be other ways of targeting that. Yeah.
And would you consider this an immune checkpoint inhibitor?
It is an immune checkpoint inhibitor, yeah. So one way to describe it is a macrophage checkpoint inhibitor. That's a concept that people can understand. That's the way I would think about it, yeah.
Great, thank you.
I think we have 2 more questions for the moment. And we have plenty of time. We're unusually running ahead of time. So.
Annette Brundle from BioWorld. I was going to ask about the analogies to the PD-1, PD-L1 blockers. Has anybody tried those with the lymphomas? And sort of in the very big picture, what's the advantage of going after the innate immune system versus adaptive? And when would you use what? Or do you think they would be used in combination? What's the really big picture here?
Right. So to date, the checkpoint inhibitors that have targeted the adaptive immune system, so T cells, PD-L1, PD-1, they just haven't been as effective as one would like in non-Hodgkin's lymphoma. So certainly they're very active in Hodgkin lymphoma, but in diffuse large B-cell lymphoma, the response rates are relatively low. And that's also been true in indolent lymphoma. So it's hard to say one is better than the other. In the preclinical models of this agent, there is some idea that perhaps T-cell responses can be induced, actually, because of the macrophages and some of the cross-presentation. So they're not really mutually exclusive. I think that is a rational target to add to this. So like I pointed out, one of the future directions is what agents should be added to this particular construct. I think a T-cell checkpoint inhibitor is a rational thing that probably is going to be added in combination studies. So I don't know that it's necessarily one is more effective than the other, but it sort of focuses on my last point of it's better to understand which patients benefit the most. And so that's one of the most critical sort of translational questions.
Hi, Nancy Melville with Medscape Medical News. Can you tell us how many patients so far have made it past 2 years, and are there any clues from them as to characteristics that respond?
So The last question, the answer is no. We don't know exactly who responds. The only ones that have any meaningful long-term follow-up out to 2 years was that first cohort in the phase 1. So there's about 25 patients. And so the ones that had good responses, I think the number is around 15 of those patients are out to that 2 year. I'll show that data later. So, but we haven't, and we've also seen that patients that had a partial response at the time of presentation have subsequently converted to complete responses. So the median follow-up of the whole group of patients that we'll show, which is around 100, is short. It's still very short because this is an ongoing trial. So we don't really have that data yet. And one other thing I wanted to point out that I didn't say to the last question was, one thing that this agent has not seen in this study is there's been no autoimmune toxicities. So that might be a potential advantage. Of course, that's a concern of the T cells as it affects other cells. We have not seen that. So you'll see the toxicity profile here when you get to the long-term duration, it really just levels off. So all the toxicities is sort of in the first month.
Wonderful. So if there are no more questions from you, I've just got one last one.
Yep.
You said CD47 was expressed widely, so you've presented exciting data on indolent lymphomas. What about other disease categories?
Right. So a lot of the preclinical models actually, so it's not unique to lymphomas. So the other multiple different cancers, this model system works preclinically. The single agent studies which have been published in the JCO was less exciting. I think some of the issue there becomes like how to place the eat me signal on these various tumor types. So I do know that this is being broadly studied across cancers, not just lymphoma. And I think any monoclonal antibody that works through an Fc receptor at least has the potential to place an eat me signal that can be sort of like work synergistically with this type of agent. So that's where I think a lot of the studies are being thought through.
But since we're hematologists,
Yeah.
if you were to choose a second disease that you were excited to extend your studies to,
Right. So, I mean, one rational thing to think about, to leads up to the next speaker might be myeloma, right? So, something like daratumumab or something like that. That would be important. One of these other tumor types, heme malignancies, that doesn't really have immune therapy that works as well as we would like. At least not the checkpoint inhibitors.
great.
Yep.
Thank you very much.
Okay.